Well, hello, everybody. So my name is Alex. I came here to present the work of the past 3.5 years and with the remainder of my PhD, which is the PASTAQ pipeline. And how can you use it to get improved preprocessing quantification for your data in LC-MS? So we published this paper last year, and PASTAQ stands for Pipelines and Systems for Threshold Avoidance Quantification of LC-MS data. The tools that we provide, they are open source. You can use them right away. Please, if you use them, let us know. Also, if you have any problems with them, hopefully you will get excited with this presentation to use them and to let us know. But the goal of these tools there is essentially to try to get better quantification out of your data, right? In the paper, we focus mostly on MDDA, but we talk a little bit about DA later. So why threshold avoiding? So we want to avoid setting any sort of intensity threshold that can remove any sort of signal that can have biological relevance. For example, if you are doing a biomarker research study, you really do not want to remove anything too early, right? You really want to get as much information as possible and then have more power for your statistics. And we also focus predominantly on MS1 quantification. This is for several reasons, but as you know, in DDA, at least the MS2 can have certain stochasticity that makes sometimes that you might miss certain peaks from appearing in your MSMS. We build this pipeline with focus on performance and trying to make it as flexible as possible. We provide some utilities for both users that are not so keen in programming, right? That's like, so you just want to use the software, do you want to use the pipeline? But if you have the power in there, if you want to get a little bit more detail out of your data. We do not, however, provide any sort of identification engine, right? So this is, you need to use your own. So we export, or you can import any kind of like MZID file that you obtain from the identification. We might support this in the future, but for now, this is not it. There has a lot of moving parts, and I'm not gonna touch in any sort of detail any of them, but you know, feel free to reach if you're interested in how these things kind of work. But the idea is that when you take your initial data, then you can convert it into a HUPO standard MZXML or MZML to then process with PASTAQ. Or you can use it to NGF, for example, to pass to identification engines, which there are many, and they serve multiple purposes, right? We created this little tool called PASTAQ GUI, which essentially allows you to parameterize and just run the pipeline right away. So the only thing that you actually need to do is just to input your files in there. You already have some default parameters so that you don't have to change if you don't want to, but you can change them. And then this also generates the parameters in the same kind of way that when we are doing the low-level programming for example, for more data science or like a little bit more ad hoc scripts, then you can use that kind of information just to load directly the output from a PASTAQ GUI into your scripts. Or for example, if you have the need to run the pipeline in a cluster, if your files are too big, which we'll discuss later. Some of the things that we provide are quality control plots. This is one of my favorites. This is the quality, the similarity matrix of different samples in a data set. As you can see, if you do not perform retention time alignment, the similarity is fairly low, and you can see that it dramatically improves when you do the processing, which it is done by default, right? Like the pipeline is ready to do like sort of like a simple quantification of DDA, hopefully with better performance. We also offer some other quality control plots, as you can see here, which I don't really have the time to get into it. But what we mean by quantification performance? So we did several essays. One of them was with like a human yeast and E. coli protein. We analyzed with both our pipeline and with MaxQuant. And we studied the reproducibility as measured by the coefficient of variation of all the peptides that were quantified, right? As you can see, in all cases, we did perform better than MaxQuant in this particular essay. We also wanted to see how, like the fact that we are this threshold avoidance approach, how low can we go in terms of intensity until we stop detecting things, or you might detect but not being able to quantify it properly because the peaks are too distorted, right? And as you can see, we still, as the intensity goes lower, in terms of the spike compound that we added, we still managed to obtain a significant number of quantified peptides. And there's another thing to be said about the fact that we are using MS1 quantification, and is that if you only quantify the MS2, you might be losing out on some potential information that you can use for another study further on down the line, right? So as you can see, both with the identifications that we get from PASTAQ, but also MAX1, we get this kind of similar profiles of the proteins for the different mixture that we get in here. But there is a large number of features that did not have any sort of identification, but they do still exist in the data. So these features could still be valuable, and you could potentially, go to the retention time and M/Z, go do some sort of target analysis for them, and then get the identification for those later down the line. And what about DIA, right? So like DDA is fine, but like DIA is pretty hot right now, and we also want to help people process that data. So essentially, we can use the same similar approaches that we use for the DDA processing. So we just treat MS1 quantification exactly the same, and for the MS2, we treat it kind of like the MS1 quantification. We quantify the peaks, then we link them together based on retention time with some ranking and so on. And this is still early on for this particular method, but we already have like some promising results. So we have a couple of files that we have done so far in which we use these publicly available LFQ bench data sets with a triple top for 32 fixed windows, which is commonly used for benchmarking this kind of application for the pipelines for pre-processing anyway. And we were mostly focused in this case about the number of protein groups quantified and the number of peptides, right? But we also want to study in the future the quantification performance also. And we are still fairly early. We know that we can do quite better, but at least we are more or less on parity with some of the existing tools that are right now on the line. So in conclusion, the pipelines that we provide, it is fast, and we could even make it faster with some more time and resources, hopefully. Hopefully it should be easy to use for people that do not want to finagle with like a lot of configuration. And it's open source. You can use it in different operating systems. We actually use it also in the university cluster for like very long analysis, because some of these files, especially on DAA, they can get huge, and maybe your computer cannot take it at that point, right? It is suitable for proteomics and metabolomics because of the MS1 quantification approach. And you can, in principle, quantify more low intensity signals. And hopefully the quality control plots also will help you find some issues with the data. It sure has helped us find some issues with some data sets in the past. So hopefully you like what you see here, and please feel free to reach out, take a look at what we have available. And if you have any questions, I will be here later. Thanks.